Hello everyone, welcome back to my Scanpy video tutorial. In this video, I'm going to show you how to integrate dataset in Scanpy using BBKNN. BBKNN stands for Batch Balanced K-Nearest Neighbors. So it is a fast and intuitive batch effect removal tool. So first let's load all the packages. We need Scanpy for single-cell RNA sequencing data analysis. We use pandas to modify the data, and we need matplotlib to plot the figures. Of course, we need to import BBKNN for data integration. So for the online tutorial, you only need those packages. But for my Jupyter notebook, I installed ESC Vino before Scanpy pandas. So I also need to load this package to get rid of all information. So let's load the packages. Next, we set the settings. For the analysis, we can use verbosity equals 3 to show the information of the analysis, and also we set the figure parameters. So now we can load in the data. I'm going to use the GSE 132771 dataset. In this dataset, they performed the single RNA sequencing for the mouse lung after bleomycin treatment, and also for healthy human lung and IPF lung. I'm going to use 3 repeat RNA sequencing data from 3 healthy donors. They named as NML 1, 2, and 3. If you want to use this dataset for practice, you can go to the NCBI website, find the GSE 132771. If we scroll down, you can see all the dataset. You can download the NML 1 or 2, 3 or. There are 3 datasets for healthy human lung from 3 different donors. So they use the same technology, 10 times genomics, performed the sequencing. So the batch effect will come from the time difference for single-cell RNA sequencing data preparation, or the sample difference from 3 different donors. So you can download the data. I downloaded already. So let's have a look at the data. You can see here we have 3 files for AML 01, barcodes, genes, and matrix. After you downloaded the data, they are in compressed form. You need to unzip them. And also, we need to keep the gene file's name as genes. For SWOT analysis, we need to change this one into features. So I have all 3 data. You can see AML 01, 2, and 3. So now we can go to Jupyter Notebook and start to load the data. First, we load the AML 01 as A data 1. Then we can make the variables name unique. This is to make sure we don't have repeat gene names. Then we do the same for AML 02 or 3. So now we loaded 3 datasets. We can use the concatenate function to merge them into one and named as A data. Because we want to see the batch effect, we named the data from different donors as AML1, AML2, and AML3. So now we have the A data. We can have a look at the A data. You can see at the moment we have more than 12,000 cells and more than 33,000 genes in the dataset. So next we need to perform the quality control. We filter the data and only keep the cells have minimum 200 genes, and also keep genes that have expression at least in 3 cells. So we filter the data. Now we can have a look at the A data again. You can see we only filtered out 26 cells, but we filtered out lots of genes. So in the A data cells were the observation names. We can look at the cells. You can see here the observation names are the cells, and the variable names are genes. We can have a look as well. You can see the variable names are genes. So next we can calculate the mitochondria DNA percentage using the calculate QC matrix function. Let's run this function. Now we can have a look at the observations in the A data. You can see the row names are the barcodes, and we named the batches as AML1, AML2, and AML3. We have the N genes. This column are the genes that before we filter out the cells. Then here are the N genes by count, total count. We have the percentage of mitochondria DNA. By default, in Scanpy, if we want to see the data, it always shows the first 5 rows and the last 5 rows. If you want to see other rows, for example, you can use the head for the first 10 rows. Then you can use the tail function to see the last 10 rows. Let's continue the quality control analysis. So we can use the wiring plot to have a look at the N genes by count and the total count and the percentage mitochondria DNA for the merged data set. You can see we generate the figure for the wiring plot. We used these 3 data sets for video tutorial in Surat, and you can see from the wiring shape, N gene by counts for majority of cells are below 4,000. And I always got a question, how can we decide how to select the parameters? So normally we keep the cells in this wiring shape. You see here, we select by 4,000. For the total count, we select cells below 20,000. And for the mitochondria DNA percentage, we keep cells below 10%. So this is a general guidance for selecting the parameters. The purpose for this, we just need to get rid of low quality cells for analysis. It really depends on your analysis. If you are looking for rare normal cell population, you probably can keep all the cells for the analysis. If we want to study signaling pathways, gene changes for known cell types, you can use more strict selections. For example, only keep cells in necessary gene, and we can use even the mitochondrial DNA percentage below 5%. After analysis, you need to find the cell types in your cell canisters. So let's use the parameter for gene count smaller than 4,000, total count smaller than 20,000, and the mitochondrial DNA smaller than 10% to subset the cells. So we can use the wiring plotter again to have a look at the cells for further analysis. You can see now the wiring shape changed. We only have cells for the N genes below 4,000, total count below 20,000, and the mitochondrial DNA percentage below 10%. And we can have a look at the A data again. You can see now we only have 11,827 cells and 20,168 genes. So we perform the quality control. Next, we can normalize the data. And in Scanpy, we also need to perform the logarithmize separately in Surat. If we normalize the data, we perform both steps. So next, we identify the highly variable genes for downstream analysis. So now we can scale the data, then perform the PCA analysis. So we can save the data from here for BBKNN integration. Because for the demonstration, I want to run normal workflow to show you the batch effect. But normally we just integrate the data using BBKNN, and the steps for quality control and the analysis until PCA is the same. So we save the data from here. Later we can load the data to perform BBKNN analysis. So from here, I'm running the standard workflow to show you the batch effect. First, we identified the neighbors. Then next, we perform UMAP analysis. Now we can use the laden method to find the canisters. So we are ready to see the cell canisters from 3 samples. We can color the cell canisters by laden analysis, and also we can color the cells by different batches. So you can see we have 2 figures down here. On the left hand side are the cell canisters by the laden analysis. We have 9 cell canisters here. And on the right hand side, the cells are labeled by their sample names NML1, NML2, and NML3. We know these 3 datasets from Surat analysis, they have batch effect. You can see here in Scanpy analysis, we also can clearly see the batch effect. The cells are not integrated very well by the standard workflow. So we need to perform data integration. After we use the BBKNN to remove the batch effect, you will see the colors from different samples mixed very well from 3 different samples in all the canisters. So let's have a look at the A data after the standard workflow analysis, you can see here in the unstructured layer, we have the neighbors, UMAP, latent clusters, and the latent and the batchy colors. In the layer of organization M, you can see PCA analysis and the UMAP analysis. So I showed you the standard workflow analysis to reveal the batchy effect among 3 samples. Now we are ready to perform the BBKNN analysis to remove the batchy effect among 3 samples. So let's read the data back we saved after PCA analysis. So we can have a look at the A data again. You can see now in the data for the unstructured layers, we only have PCA. We don't have the rest analysis for neighbors, clusters, and also in the observation M layer, we don't have the UMAP information. So for the BBKNN analysis, you will need to do the same as load the data, perform the quality control, normalize the data, and perform PCA analysis. Because we saved the data for those analysis, we don't need to do it again. We can directly do the BBKNN analysis now. Use the BBKNN function, and we set the batchy key as batchy. Then we can remove the batchy effect among 3 samples. Let's run the BBKNN function. You can see it is very quick. So after remove the batchy effect, we can perform UMAP analysis. And run the latent function to find the clusters. We use very low resolution for the purpose of video tutorial because it is easier for us to label the cell clusters. So let's run the latent function. Then we can have a look at the cell cluster again and count the cells by the latent cell clusters and also count the cells by the batches. Let's run. You can see now we only have 8 cell clusters among 3 samples. The most importantly figure here for this video tutorial, we can see after the BBKNN analysis, we removed the batchy effect among 3 samples, and you can see now here the cells were mixed very well in each cell clusters. So now the data are good for other analysis. So let's identify the cell types. We know in the lung we can use 4 cell markers: PTP-RC for the immune cells, EPCAM for the epithelial cells, CLDN5 for the endothelial cells, and collagen 1A2 for mesenchymal cells. At the moment, we don't need to go to the details for the immune cells, epithelial cells. We can identify the cell clusters by the marked genes as 4 cell types. So first, let's run the mark gene for the PTP-RC to identify the immune cell clusters. You can see PTP-RC label cluster 0, 3, 6, and 5. Then next we use EPCAM to identify the epithelial cell clusters. You can see now EPCAM labels cluster 2 and 7. We use CLDN5 to identify the endothelial cells clusters. You can see CLDN5 labels cluster 1, And finally, we use the collagen 1A2 to identify mesenchymal cells. You can see collagen 1A2 enables cluster 4. So now we know the cell types in all the cell clusters. We can name the cell clusters. We know cluster 0, 3, 5, and 6 are immune cells. Cells in cluster 1 are endothelial cells, 2 and 7 are epithelial cells, and 4 are mesenchymal cells. So we can rename the cell clusters. Now we can have a look at the cell clusters again. You can see we labeled the cell clusters as the immune cells, epithelial cells, mesenchymal cells, and endothelial cells. So now we can save the data for future analysis. Next time, if we want to use the data to perform the analysis, we can load the data back into Jupyter Notebook. We can check if the cell clusters are the same or not. You can see we saved the data correctly. Okay, so that's today's video demonstration. I showed you how to load the 3 single-cell RNA dataset from 3 different healthy donors and performed the quality control, removed the batch effect, used BBKNN, then performed the dimensional analysis and clustered the cells. We also used the mark genes to identify the 4 cell types in the human lung and renamed the cell clusters. We saved the data for future analysis. You can see the BBKNN is a very quick, easy method to use. I hope this video tutorial can help your data integration in Scanpy. If you like my video tutorial, please subscribe my channel if you haven't to do so, and share my videos with your friends. Thank you, and see you in my next video tutorial.